From Microbe TV, this is TWiP, This Week in Parasitism, episode 158, recorded on September 13th, 2018. I'm Vincent Racaniello, and joining me here in New York City, Dickson Despommier.
Hello, Vincent.
And remotely, Daniel Griffin.
Hello, everybody. Welcome back.
Daniel. Hello, Daniel.
Yeah, we are doing a quick turnaround here, right? 2 weeks?
We are.
And Daniel, the weather out there is as weird as it is here?
The weather, I'm hoping people are starting to use the terminology of climate change and global climate chaos.
That's right.
It's chaotic.
Because how can you not believe that's going on when you when you live it?
Exactly. If I were a trout, I would be so confused right now. I wouldn't know what time of the year it was. I wouldn't know what to do next because the water temperature goes to 70, which is horrible, and then it goes down to 50.
Did you go fishing this week?
No, but within 3 days that happened. That's crazy.
Well, I turned the heat on Monday, and now you don't need it anymore.
It's weird.
And
it is weird.
It's been raining for essentially 2 weeks, right?
And there are places that are listening to this and say, I want a drop of rain. I don't care how much. Just bring me rain.
It's really foggy because it cooled down and now it's warmed up.
Well, exactly.
Anyway,
and then we got this hurricane bearing down on us.
Well, it's bearing down on the Carolinas, right?
Yeah, we're going to feel it, though.
What are we going to feel?
We're going to get rain.
Daniel, do you have to batten down your boat?
No.
And the hatches. My youngest brother has a house in Wilmington, North Carolina.
No.
And I feel that in the floor of his house is 15 feet above sea level. So I'm feeling like that floor will be underwater tomorrow afternoon.
If he has a house.
If he still has a house left. And I think
that's a direct hit, isn't it?
Shed somewhere. That's where it's going to hit. That seems to be the bullseye.
Tomorrow is the landfall, is that right?
That's right. And it's going to be slow, so it's going to dump a huge amount of rain. They estimate 30 inches of rain in some places. Yeah. That's crazy.
Yeah.
Sounds like you're living in Houston.
All right, well, good luck to everyone, but this will encourage parasites, right, Dickson?
Eventually. Katrina did that, by the way. We can talk about that later, by the way.
All right.
It did.
Let's go to our case from TWiP 157, Daniel, back to Thailand. I was going to say Thailand. My thighs.
Thailand, that's Radio City Music Center.
Yes, back to Thailand. Let's not make fun of people with speech impediments.
No. Just a different part of the anatomy, that's all.
We presented a case of a housewife, a woman who stays in the home and is a homemaker. And she initially was seen with a complaint of red bumps on her body, described intense itching, saying that was worse at night. These bumps were on her head, her legs, her upper chest. But she also reported that additionally, she didn't feel well. She felt feverish. She had muscle aches, some difficulty breathing. And we got a little contextual information about this. All began a few days after she, her husband, the family moved into a residence that had been previously abandoned. And when they moved in, the residence had coconut husks around. The roof was thatched. Later on, the husband and children started to develop similar symptoms. They also found some unused mattresses, which had been actually constructed from the coconut husks. There were some rat nests around. No other travel, no other pets, no other animal exposure. And on exam, though she wasn't febrile, didn't appear toxic, she did have a little bit of an increased heart rate. She was having a little bit of difficulty breathing, and she had multiple red papules with this central punctum. Her labs were unremarkable, except for, I guess I'll say her differential was normal with only 6% eosinophils within the normal range. A skin scraping was done from one of the lesions on her chest, and I suggested that was what helped us with the diagnosis.
Okay. We had a few guesses. First one from Alexander, who first writes, I think I should clarify that the tool I mentioned in my previous email is a cruiser's stick, which uses a law of similar triangles and your arm length to be able to measure a height and diameter. They have two parts, sometimes a third being a yardstick, a hypsometer, and a Biltmore stick. To determine heights, you can walk back a set distance and see the height as the top of the tree should fall between lines marked on the stick. It isn't considered accurate, but when you are 66 feet away from a tree and guessing where the top of it is among the forest, it doesn't really seem to matter if you're a few feet off. Then again, it does have its limitations. It doesn't really work on slopes and shouldn't be used for cruising high-value timber. Does anyone know what this refers to? Alexander talked about this in a previous email, he says. Nobody remembers, huh?
I'm lost.
Oh.
Let me see if I... Oh, here it is. Well, I was learning about forestry. This is from previous TWiP. My professor always stressed the importance of having something to fall back on when newer techniques don't work. I remember hearing that with the old aerial photography, you could actually count each individual tree and learning how to measure structures from the same photography. I have a yardstick that I can use to measure diameters and heights of trees without having to use any specialized tools. And I think we probably said, how do you do that?
Right.
So there you go.
Yep.
Now, on to the case. I had initially thought it might be scabies with the nocturnal itching, but the primary infection are delayed by several weeks and the rash doesn't really sound right. Next, I thought of bed bugs, Cimex hemipterus, but after further reading, I saw paired bite marks, and recalling the unused mattresses mentioned in the case, I didn't want to get stuck on it. Looking further at parasitic diseases 6, I went to fleas, which I thought about because of the rat nests present and how they moved in recently. This is going to be a guess, but I think that when the family moved in, they might have scared the rats away, and without food, fleas might have taken the opportunity to feed on the family. I'm going to stop there and say that my diagnosis is going to be that they might have some fleas, which may have given the woman a secondary bacterial infection. May I be so bold to request that in the next edition of Parasitic Diseases, you make a diagram of differences in bites and throw a few drawings on it, as while looking for a description of the bites, I only found one website that said they had a single central puncture, which is mostly all of what I'm basing my diagnosis on.
Right. Well, the difficulty of that, we've discussed the possibility of that, is that it's not always true. That is to say, if they bite in twos, the insect could be biting in ones, for instance, or threes, and then you can get thrown off. So there's nothing absolutely written in stone that's the way it presents. But if you see enough of them, then the statistics favor one or the other. But I appreciate the suggestion. Chris writes, hello, professors. It is a cloudy 70 degrees F here in Stony Brook, Long Island. For this week's case study, I believe the woman and her family are suffering from bed bugs. This was a much easier one than the last 2 or 3 cases, so I feel pretty good about this. One holdup I have is that Daniel said that the way they solved the case was to do a skin scrape. Well, yeah, right. I was under the impression that bed bugs stay on the bed, so why would a skin scrape work? Do a few of the bed bugs not jump ship when the woman gets out of bed? Another possibility is that she could have had scabies or even fleas, but I don't think these 2 options are as viable. Again, please don't put me in the running for the book, as I already have one. Also, you mentioned you found the paper about ant parasites. One of your listeners sent in. Interesting. So I figured I would send in relevant work. This work focuses on a trematode that also has a host manipulative behavior that varies depending on the temperature and humidity. And being that this is the week in parasitism, thought you would all enjoy in addition to the paper sent in last week. And he lists a reference.
And in fact,
guess what we're going to do?
This is related to the paper we're doing today.
You bet. I think it is a really very cool and classic example of host behavior manipulation that is commonly observed in the animal world, and that it would pair well with the paper sent in last week. Lastly, I have a question for Daniel. How do you deal with the ticks on Long Island? As I have previously mentioned, I recently moved to Long Island, and I knew that there was going to be a lot of ticks. But last week, I found over 60-plus ticks on me. I lost count after 60. It was probably closer to 100, though. I acquired all these ticks after about 10 minutes of walking through a nature preserve. The ticks seemed to get stuck in my shoes and socks, so I ended up putting my shoes in the freezer for 30 minutes. And then I went to my lab and had to spray my legs with 95% ethanol to try to wash off the ones remaining on my legs. Is this an effective strategy for when I inevitably get swarmed again? Once again, thanks for everything. I recently started to ride my bike to school every day, and TWiP and TWiV make my 40-minute ride a very pleasurable time. Warm regards, Chris.
All right. So we'll get back to the ticks.
Okay.
Right. Because otherwise we get to talk about that a lot. So let's, Delbert writes, hello, TWIPsters. My guess for the case presented in TWiP 157 is that the offending parasite is bed bugs insects. The two most common species implicated in human infestations are Cimex lectularius or Cimex hemipterus, the former of which is cosmopolitan and the latter being found mostly in the tropics or subtropics. Like other members of the Hemiptera order, they contain piercing, sucking mouthparts. Mm-hmm. It sounds like the evidence presented by Dr. Griffin that these little parasites have infested the coconut husk mattresses or have taken up residence somewhere else in the patient's new home. The fact that the rest of the family is also suffering similar symptoms points to bed bugs as well. While a nuisance to have in a home or dwelling, they are not usually of any severe medical importance. Their primary medical importance is inflammation associated with their bites. While some Cimex species have been found to harbor blood-borne pathogens, they are not effective vectors of disease. Treatment includes over-the-counter medication to relieve itching, cortisone creams, and antihistamines. Excessive itching, scratching can lead to secondary infection. Treating the house to rid it of the infestation is also possible, however expensive, and probably not within the means of the family. Please keep up the good work with the podcast. I am a longtime listener of TWiP. I started listening in grad school while I was still a PhD student. I've listened to every TWiP episode, and I am now an avid listener of every other Twix series. I listen to all of them all the time. I subscribe to all and never miss an episode. Do not stop doing these. They are immensely useful and helpful. As a matter of fact, early TWiP episodes that cover the basics of parasites are required listening for my parasitology course. Also, recently, I've switched over to my course book to parasitic diseases, particularly now that it is free and will not use any other textbook. Thank you immensely for making that book free and accessible. There are a lot of colleges, universities out there where students cannot afford textbooks. You are doing a great educational service. Fantastic work. Thank you, Delbert.
All right.
How about
nice,
that?
Delbert. Thank
Very
you.
Nice. Yep.
Adil writes, Dear doctors, firstly, thank you for your TWiP smart banter and utterly engaging podcast. After multiple weeks of being right and yet reticent to take my shot, I will hazard a guess. Initially, I thought scabies because the itching was noted to be worse at night. The punctuation and explicit reference to unused bedding made me think of body lice, however. The regions impacted seem to align with this as well, and like with last episode's diagnosis of babesia, I am not at all confident, but there it is. Hope all is well with you and that I can send forth future responses with a bit of conviction in my diagnosis.
Right. Kevin writes, cool, like autumn. Our case involves a woman, in parens, and her family with an unfortunate dermatologic affliction. The itch, rash, scruff, pruritus, cooties, dermatitis, eczema, prurigo, lichen simplex, the eruption, the crust, however described, a widespread pruritic rash can result in anguish of an almost biblical magnitude. It was often taught that the skin was the largest organ in the body. Is that still true? Now, there is an immune system, the biome, gastrointestinal and otherwise. Nonetheless, considering weight and surface area, the skin is still a contender. It is very sensitive, full of nerves, and raises the alarm when invaders are about. Acute cutaneous eruptions or symptoms may be due to a primary dermatological condition or an external manifestation of an internal condition. The tropical location, presence of a known substrate for arthropod pests, acute onset, cohabitation with rodent pests, and multiple afflicted family members strongly argues against a systemic disease, i.e., chronic renal failure, cutaneous lymphoma, cholecystatic pruritus, rickettsial infection, scrub typhus, etc. The papular nature of her diffuse rash, along with central punctum, argues against bacterial or viral skin eruptions, such as erysipelas, impetigo, herpes, furunculosis, etc. This leaves us with whatever hath more feet among all creeping things that creep upon the earth. I will discount helminthic infections such as Toxocara, Strongyloides, hookworm, etc., as inconsistent with our case. Likewise, we may forego consideration of biting flies and myiasis, cutaneous maggot infestations with Cymex lectularis. Bedbugs is a possibility, but the widespread nature of the rash and intensity of itching argues against this. The fact that the home was vacant for several weeks prior to the patient's occupancy raises the possibility of a flea infestation. To quote the Gordon and Lavopierre book, this vibration-induced hatching probably accounts for the many instances of people who have entered deserted dwellings and later attacked by large numbers of fleas seeking their first blood meal. End quotes. Flea bites are less likely to produce such widespread rash, and the patients would probably report seeing these fairly visible insects. Smith's Medical Entomology text, 1973, in a flight of terminological excess, states, a flea puncture is characterized by a tiny dark spot, purpura pulicose, surrounded by a patch of swollen and reddish skin, roseola pulicose. The presence of rat nests, besides being a risk for flea infestation, prompts discussion of ornithodosis, Bacoti, the tropical rat mite, a hermatophagous arachnid that can cause dermatitis in humans. These mites have been found in gerbils and other rodent pests in Europe. See references. Diagnosis requires a microscopic examination of the mites. Of course, scabies should be mentioned, though the abrupt onset of the dermatitis, coincident with the moving into an environment conducive to other male mite pests, allows me to dismiss this idea. Coir, a fiber obtained from the coconut husk, is used in a variety of manufacturing. Coir mattresses are very popular in India, see the reference, and the tropical world. They are also widely available in the U.S., with one king-size coir mattress retailing for $4,500. That's a lot of money for a mattress. Our patients moved into a home that was presumably a component of a cottage industry producing coir mattresses. The large quantities of coir fibers in the home makes the diagnosis of copra itch very compelling. Note, copra is the dried meat or a kernel of the coconut. The PD6 text does mention skin irritations due to mites associated with straw, flour, grain, dried fruits, vanilla, copra, and cheese. These conditions often are colloquially referred to as grain itch or grocer's itch. A case report almost identical to TWiP 157 from the Moriang area of southern Thailand is beautifully described by Sarathep in the journal ID Cases 2018 open access reference below. The mite Tyrophagus loquior causes copra itch and an intensely pruritic dermatitis that results in widespread erythematous papules and can cause systemic symptoms such as myalgias, fever, shortness of breath, and fatigue. Sarathep also provides a clear photograph of a papule with a central punctum. These mites do not tunnel under the skin like Sarcoptes, but bite and attach, as evidenced by Sarathep's photo. A photo of Tyrophagus recovered from a papule. Treatment with 5% permethrin cream is advised. Adjunctive topical steroids for the most pruritic areas and oral antihistamines for generalized pruritus may also help. The entire environment must be thoroughly decontaminated and all of the debris and rodent habitats removed. Thanks to TWiP team for stimuli as piquant as the bite of a mite. And he has a bunch of references below, which I will not read, or perhaps at this moment cannot read.
Dickson, you have a coir mattress?
I don't. No, I have a Tempur-Pedic mattress.
All right.
That's what I have. So those are the guesses.
Those are the guesses.
They are the guesses.
In fact, we had 5, of which 3 will be in the running because Kevin and Chris
have books.
They got books already.
That's amazing.
So
you could run and give it to a friend, but that's
some
okay.
Interesting thoughts here.
Very.
So, Daniel, what would you like to do first?
Well, I think the first thing is I'm curious what you two might be thinking. Shall we start with Dickson? Dickson, what were your thoughts, or what do you think salient aspects of the case were?
Yeah, well, my first guess, minus the skin scraping, was fleas, because I do know that there are incidences, and I know someone who actually had this happen to them when they went on vacation and rented a summer home from somebody who had a pet dog. And when they walked in the house, which the dog hadn't been in 3 months, these 4 people were attacked by fleas that hatched all over the place. But you can see the fleas, right? And they're ectoparasites, and they're not, you don't do a skin scraping to find out if the bite was caused by a flea or not. So then when you said it, the diagnosis was helped along by a skin scraping, I thought about scabies, but scabies is not a parasite that survives like this. But I did think about bird mites, Ornithodorus and things of that sort. But I think these core mites associated with the Thailand-derived copra from these coconut shells is probably the right thing to say here. I would agree with the last diagnosis.
Yeah, I thought scabies, but I didn't like it, but I didn't know about these mites at all.
Right.
I had no idea.
Right. And our book doesn't go into that kind of detail.
Why is that?
That's a good question, actually, because I don't think that they're that common. I think that they're very uncommon, all right? At least where we are.
Yeah, that's interesting. We don't, actually. And it is, occasionally people will comment about something we refer to as a minor parasitic disease. And for instance, when we live in Long Island and we put babesiosis under minor parasitic diseases, most of us here say, how could that possibly be? Because for us, and then you look at other ones that we don't really spend much time on, where hundreds of thousands of people are suffering from them. And it really is a challenge what we're going to focus on and what not. And I don't know if we spend as much time on ectoparasites as perhaps we should. And as the person mentioned, if it turns out this is one of those mites, we do discuss that in different areas of the world, there's skin irritation due to mites that are associated with straw, flour, grain, vanilla, copra, cheese. And I like your thinking on this. This is it sounds a lot like there's some sort of an ectoparasite involved.
Yes.
We had one of the people write in, I think, hoping that in our next edition we could provide enough information that you could almost use the rash that you see as part of a differential to guide through an algorithm and say, oh, here we're seeing these linear burrows between the fingers. Boom, that makes you think this diagnosis. We're seeing 3 bites in a row. That makes you think this other one. And Dickson, you're definitely correct. And I think you don't want to provide information that is untrue, even in today's world. And
oh, why not?
Even when
Let's just do it.
Becomes popular. And they're
fake bites.
Well, I was discussing a case today, and I was saying, yeah, we all have been taught this, even though it's not true and hasn't been true and continues not to be true. But don't worry, that doesn't stop people from teaching it.
No. And
people love to teach with the bed bugs. You see 3 bites and just think breakfast, lunch, and dinner. Now that's not true.
No,
but it's a wonderful mnemonic, and people seem to want to see that.
Right.
And so I think we have to be careful when we put such information in our book. And in this case, there were a few features that I think were helpful. Part of it was the epidemiology. Part of it was where the bites were, that they were not in specific places that we might associate with scabies, for instance.
Exactly.
And I hope I gave a pretty good hint by talking about the coconut husks and the mattresses there. And then even the hint that the skin scraping got it, because as our emailers correctly mentioned, bed bugs are not going to stay and be identified in a lesion like that. And when the scraping was done and looked under the microscope, there was an 8-legged little creature there, and it was actually identified as the copra itch mite.
Cool.
Yeah.
Very cool.
So
how long do those little things remain alive without a host?
That's the interesting issue because there was that period of time. And I actually was thinking about that as we were reading the emails about, if you leave a place long enough, will the bed bugs be able to survive without some sort of warm place to go and feast? And I don't know what the mite lifespan is without human beings, but there were rats there as well. So
that's true.
They have had potential.
So they're not picky. Or if they are, they don't get picky when their preferred host is not there.
Yeah, I don't believe that the coir associated, the copra itch mites, are restricted to human beings. So maybe we'll get one of our emailers who might be a copra itch mite. But my understanding is they can go to dogs, they can go to rats. My understanding is that they actually can go to various other other animals, mammals.
Right.
The other thing I think is interesting about copra itch, and there's a lot of mite infestations. I like the fact that someone used that terminology. There's a lot of different mite infestations in different parts of the world, and I do actually think this is an underappreciated disease. There's been a lot of issues. The British describe this. It's great actually to read some of the early descriptions when they were in India. And they started running into what is this, and actually isolated these, and they had the microscope to be able to visualize them. And so there are different mite infestations that we see in different regions associated with different, we'll say, industries, so different plants and harvesting and production.
That's true. And some of them, surprisingly, can transmit helminth infections. There's one that transmits a filarial parasite of rats called Despommier carinii, and it's transmitted by a mite. And when you look at a mite, you say, how the heck can you squeeze a worm into a little arthropod like that? And the answer is you can.
Yeah.
The microfilaria are very, very small, and I believe it's a dermanicis mite, but I'm not. I don't want to go on a limb on that one, but I know that I knew someone who actually worked on that parasite for their thesis at the University of North Carolina. They worked on Despommier, and they talked a lot about working with mites, and they're very difficult to work with because you can't see them. They're really invisible little beings that you have to do a microscopy for in order to get them. And I'm not pretty sure a PCR test would not have turned this one up.
Yeah.
A lot of parasites are now PCR based, but I don't think we're going to get a PCR test for mites yet.
There's even a tick that transmits a filarial disease. Go back to our person who was asking, what do we do about ticks in the area? Exactly. But no, the ectoparasites are a major issue, and we're talking about the mites here. But scabies was so endemic in certain areas, and I think I mentioned some of the time I've been in Panama, where you go into communities where 20% of the population has it. Some areas of the Caribbean, the people that had the thickened, rough skin from ectoparasitic infestation and scabies and that were important to do certain parts of the religious rituals because they had the ability to hold on to things because they had this really rough skin. So it's actually amazing how much time we've spent living with ectoparasitic infections and infestations.
I remember also at the end of the Korean War, a lot of Korean veterans adopted children and brought them back to the United States. And these were mostly orphaned kids. And almost 100% of them came back with scabies.
Yeah.
That they had to be treated for that.
So, Daniel, what did you see in the skin scrapings?
So the skin scraping, you could actually, you're actually able to see the adults under the microscope from the skin scrapings.
Wow. Cool.
It was actually, another feature of sketch was interesting was the systemic symptoms. Often people come in and it's just a skin, but if you actually go through the literature and you look, the description of feeling crummy, low-grade temperature elevations, and particularly the trouble breathing, the majority of people that are diagnosed with copra itch actually report the dyspnea, the systemic symptoms, which is, I think, interesting.
Do we know why? Is that from the mite or from the copra?
I believe it. So I believe it's an inflammatory response to the mites, not from the mattresses, and that brings up that wonderful thing. As noted, people are paying thousands of dollars for these mattresses, which are now quite popular in Scandinavia. And now coming to the United States. So for thousands of dollars, you can get one of these mattresses, raising that really important question of do you now have to worry? Is this going to spread to Europe and Scandinavia and the U.S.? And they process these quite a bit. So by the time they get to those places, you're not seeing, or one would not expect to see the copra itch.
Do you know if it was that mite, the copra mite?
It actually, it was the copra.
Okay.
Shall I try to pronounce it? The Tyrophagus longior mite.
Right. And how did you treat her?
So as one of it was actually the last emailer kind of went through. So a big thing was cleaning out the environment. She actually received some permethrin cream, antihistamines, and
just fine after that. Systemic symptoms went away.
And in general, that's the response that you tend to see. Once you remove this stimuli, once you remove the mite infestation, then people do quite well.
Okay.
Right.
All right. Now, Daniel, what do you do about ticks on Long Island?
Exactly right.
I like the idea of putting the sneakers in the freezer, and
right.
It is really an issue. We have an incredible number of ticks on Long Island, and this is, I think, for probably people not from the Long Island, probably shocking to hear that this gentleman spent 10 minutes and came back with 60 ticks, and I bet he didn't find them all. And I have a photo that I took. I actually was taken. I was in South India with like a bunch of Norwegians, and I don't know. I was in one of those moods where I tucked my pants into my socks, and we did a photo, and we called it Lame Disease, and it was a sort of a joke about Lyme disease. But one of the problems is the nymphal forms of the ixodes, the deer tick, black-legged tick, are so small they're the size of a poppy seed, and they are on the grass, and they are questing, and they can actually jump. They can leap. You don't need to actually even make direct contact, and contact.
We knew what you meant.
Okay, contact. And when you go into one of these areas that is heavily tick infested, they will get on you. They're tiny, so it's very hard to see them all. And if you don't have your pants tucked into your socks, like I'm showing in my Lyme disease photo, what they will not just stay there on your lower leg. They will actually migrate upwards into the dark, moist, private parts, and they'll often be there. And that's when you find them a day or two later, when they have now enlarged in size as they become engorged with blood. Wow. So you could follow the several approaches. This one is the approach that my cross country coach from high school used to take, which was that when he wanted to enjoy the out of doors, he would crack his window, and when he was done, he would close it. That was his tick exposure.
Stay inside. Yep.
So the other is to use physical barriers, as I mentioned. When you go out, to tuck your pants into your socks. And this is actually reasonable. I must say that's something I do when I'm in a lot of these really heavy tick infested areas, because they can only go so high. They're usually not going to jump from the blades of grass or from the plants to your scalp, but they will. Your whole lower body is within their range. And I guess if you're going to lay down in the grass, you're going to have some more risk. The next is you can use certain chemical things, and I think I've mentioned picaridin on prior episodes. There's a little bit of controversy whether picaridin or DEET are more effective for tick prevention. I tend to recommend picaridin because it's more well tolerated. And then the tick check, as our emailer was describing, is if you can remove all the ticks within 48 hours, it takes a process of 48 hours for the tick to transmit at least Lyme disease. But that doesn't cover everything, because now in Long Island we have Lyme disease, we have anaplasma, we have Rocky Mountain spotted fever, so even the dog ticks are an issue. We have babesiosis. The number of tick-borne diseases just keeps increasing.
Ehrlichiosis.
Yeah, so we have Ehrlichia chafiensis and Anaplasma phagocytophum. Yeah, this is the list is going to grow. Where actually we recently have had the tick, the Lone Star tick that carries starry southern tick-associated rash illness.
Why is this increasing?
Well, I think you could just take one guess at this one and say that the climate has favored a warmer regime.
But the hosts of ticks are rodents in the woods, right?
Well, no. There are two. The deer tick has two. One is the deer mouse,
right,
and then the other one is the deer.
Yeah.
So the nymphs feed on the deer mice, and then the adult ticks feed on deers. And the nymphs feed on humans as well. So
are these animals increasing in number?
Well, the deer, let's go to Long Island. Ask the gentleman who lives on Long Island. Daniel, how many deer have you seen on your way to work every day?
Well, this weekend it was the beginning of the high holidays
yes.
for those of Jewish faith, and my family celebrates these holidays, and we were out with my family out on eastern Long Island, and at one point we were driving on the way to Temple, and there was a field. And I said to my family, I feel like I'm in Yellowstone, and this should be a herd of elk before the wolves were reintroduced. Exactly. There were dozens of deer.
That's right.
And so the deer population has exploded. There's plenty of mice, and the tick population has expanded as well.
Right.
And not only that, but we're actually seeing, I think the last time that Lyme has now been reported in at least all of the lower 48, maybe all of the 50 states. It's no longer restricted to the banks of the Connecticut River. It has spread throughout Long Island. Babesiosis has spread from Nantucket, is now all throughout Long Island, Massachusetts, Rhode Island, Connecticut. And it's a combination of factors, changing situations where we have an explosion of the deer population, we have increasing numbers of rodents, but also we're seeing the ticks themselves. There's a spread of these infections throughout them.
So there's no predators either, and there's no hunting.
Yeah.
So, there's no check and balance anymore, right? And your mother, Daniel, should be appreciative of that because obviously, as you mentioned before, some other time on our show, I'm sure that your mother was responsible partially to reintroduce wolves into Yellowstone Park, and it completely reoriented the ecology of that place back to the way it was, which is the way it should be. And if you could introduce wolves out into Long Island, besides the two-legged variety, it would be fine.
I'm sure they would love that.
Oh, yeah. No, you can't do it. So they've been working on all kinds of crazy schemes to try to dart the female deer and make them sterile and cull the males so that they can't mate. And it's all for naught. There are people that actually feed the deer.
Well, it's a challenge because the deer are critical in one part of the life cycle, which is where the male and female ticks come together. All the other stage transformations, I'll call it, occur feeding on rodents. But if you kill all the deer, and they've done experiments with it, dramatically reduce the deer population. As long as there's one deer left, you may make it worse.
That's right.
If there's one, then the chance that a male and female tick are going to meet because there's only one deer, deer is going to be covered in ticks, and the ticks are going to have the chance to mate. So you would need to completely get rid of 100% of the deer. Or have an alternative approach.
It happens on certain places like Nantucket, for instance, or Block Island, where they used to have deer. And in some cases, they've gotten rid of all of them because it's a small place and you can herd them up and get them out of there. But it's the way life is.
Well, we have an increase, too, where I live in New Jersey.
Sure.
I've noticed in the years that we've been there. In fact, this summer, had lots. We had a family. We had 3 baby deer born, and the parents were there, and they're always either in the front or the back. And I come home at night, I come in the driveway, they're standing on the lawn. I walk down to get my mail. They're 20 feet away. They look at me and they go back to chewing the grass.
Exactly.
I mean, so they're not afraid anymore.
They are now peri-domestic animals. That's right. And it's why the car rates are so high. Insurance.
And I look out the window in the morning, and they're sleeping in the Pachysandra, It's so funny.
Well,
yeah, we were up in Canada recently, up in the Mount Tremblant area north of Montreal, and we were walking back towards the car. It was probably 8 or 9 at night, so it was dark, and there was a a fawn, is little baby deer. And I saw it, and I called out to my daughters and my son. I don't think my son responded, but my daughters did, and they came over. And Eloise, who's about 6 foot tall and 15 years old, she started to approach the deer. It was basically standing 2 feet away from me. And as she approached timidly, the little deer started to go towards her to basically check her out, and she ran off. She ran off. Yeah. And initially I thought that was very entertaining. To my wife pointed out that probably was wise, because where were mom and dad? And I was like, yeah, you're right. As friendly as they might be, I'm not sure how friendly mom and dad would be if you were being nuzzled by a baby deer.
Dad, not necessarily. They don't stick around afterwards, but the mother deer gets very protective.
Well, a few years ago, our dog had an encounter with a deer right outside the house, and the deer was swatting it with her hooves, and if she had hit, she could have killed the dog, right?
That's right.
But the dog put her 2 front paws on the deer's chest and was pushing her up.
Oh, really?
Silly dog.
and the deer ran off, but
good
it's pretty weird.
Yep. Well,
you just think, why do you have to mess with them?
There's a whole ecology of peri-urban pests and things that we've introduced or encouraged by our own behavior that
what I see very frequently, foxes.
Sure.
They're incredible. We never used to see foxes.
Vincent,
now we see them all the time.
You used to have fox hunting in your area, and they don't allow that anymore. It's banned, all right, because it's a cruel and unusual treatment of an animal. So they do
allow some deer hunting in our area.
Yeah, they do.
It's controlled certain times of year. It's within preserves. You can't just do it anywhere.
That's right. New Jersey has a bear season as well, so that's very controversial also.
But it's
crazy stuff.
Getting very wild.
Well, they're integrating into our society in ways which cause us pain and suffering. So
I'm reading this book by David Quammen, Song of the Dodo, and he goes through many animals that were made extinct by humans.
Yes.
Including dodos, which they were eaten on this island where they lived. Yep. And there's another animal in Tasmania. It's not the Tasmanian devil, but it's related.
It's the wolf, the aardwolf.
Yes. And they were there first, and then settlers brought in sheep and cows, and the cows start to be eaten by these. So government paid people to kill them, and they wiped them out. And he says it's funny, a few months after there were no more, the government declared them an endangered species.
Well, the last one died in a zoo.
Yes, they've died in zoos. But he thinks there's still some there. But
Thessalians, they're called.
The Thessalians. We just wipe out species. We don't care.
The greater auk, we killed all those off. We killed off the passenger pigeon.
Moas, too, right?
We're very good.
In New Zealand, right?
We were quite good at a lot of this.
All right, let's give away a book.
Good idea, Ollie.
We just had 3, right?
That's it.
So let's get a random number between 1 and 3.
There's no time for a drum roll.
Generate the number 1. number 1 is, who is number 1? Alexander.
Great.
Who measures trees with a stick.
So we have some signed books, because fortunately when Daniel was here last, we both went over and signed some books.
Yes, I have a pile here. I brought some home. I shipped some out. And Alexander, send us your address at TWiP@microbe.tv. If you are outside the U.S., which I don't think you are, include your phone number. But if you're inside the U.S., it's not necessary. Thank you very much. All right. Next, we have a paper.
We do.
Which is sent in by Hannah, who wrote, this just came across my newsfeed, a recent paper in Scientific Reports in which researchers managed to image Lancet liver fluke brainworms in situ in an ant's brain, showing the physical connection between the parasite and the intermediate host's brain tissue. Right. Here's an article, press release from the Natural History Museum in London, where the work was done. Love your show. Keep up the good work. So Hannah sent this in June. I thought we would do it. It's from Scientific Reports, which is the open access version of Nature papers. All these journals have at least one open access journal, so they can say, look, we're doing open access, but everything else, of course, is closed access.
Yeah.
Nevertheless, 3D virtual histology at the host-parasite interface, visualization of the Master Manipulator, Dicrocelium dendriticum, in the brain of its ant host by Martin Vega, Garbu, Ahmed, Wickling, Goter, Caldwell, and Hall. These individuals are from the Natural History Museum of London, the University of Alcalá in Spain, natural history, the University of Lethbridge in Canada.
Yep.
And these are
Alberta, Canada, by the way.
Alberta, exactly. And that's where, in fact, where these ants were collected in the study, and then they were shipped over to London. The first sentence is wonderful. Animals infected with parasites often behave differently relative to those that are not. And they go through some great examples. Terrestrial crickets infected with hairworms that go into water. Remember we talked about that?
A long time ago.
When you and I were on TWiP?
Yeah, that's right.
We're no longer on TWiP.
No, we're not.
Roundworms that chauffeur ground-hugging ant hosts into the tree canopy where they wave their conspicuous red abdomens to mimic edible berries. You can imagine what would happen to them.
Good heavens.
They get eaten. Fungi can infect ants and enslave their hosts to die in the precise locations on a plant that make them most likely to transmit spores. So lots of examples, right?
Et cetera, et cetera.
How does it work?
Exactly.
And in this page, in this paper, they looking at ants that are infected with larvae of the trematode, Dicrocelium dendriticum.
That's right. The Metasicaria.
And Dickson, do you know anything about this?
Well, I do, actually. I mean, I did notice also an incredible omission in our textbook that I don't think I could find Dicrocelium in our index, and I think we have to correct that.
Is it in the book?
And I don't think it is. I don't think we mention it, but it might be mentioned.
Do these infect people?
They do.
Which species infect?
Very well. Dicrocelium dendriticum infects people. And it has a broad host range. It infects cattle.
How do we get it?
Humans. We accidentally ingest an infected ant.
Oh.
How do you do that?
Lettuce.
Well, that's one way, but cattle ordinarily feed at a certain level, okay? And this parasite, this is a fantastic portion of its life cycle. It actually induces the ant. It has pincers, right? Pincers, I should say. Its mouth parts consist of pincers. Mm-hmm. To rapidly open and shut, rapidly open and shut. That's its behavior difference. And it grasps onto objects higher in the environment than most of the other ants, like a flower. Uh-huh. And that's how they collected these ants. They actually collected the flowers. The other ones were all down in the ground, right? What they were looking for were the ants that had this infection. Yeah. And so they go to a higher level and then they grasp onto that higher, whatever that is, a blade of grass, a flower, for instance, of like clover. And then the cattle come along and they graze and they eat that right up. And then that's how they get
right? Accidentally.
Well, yeah, of course, it's all accidental. And humans don't just go around eating flowers all the time. So it's a rare occurrence in humans.
You know why I say accidental? Because
it's inadvertent.
It comes along, but we eat meat. We want to eat the meat. Sure. I guess it's the same as eating grass, right? It's all accidental. You're right. This is a stupid thing to say.
Well, what they've done is selectively, the selection was to take advantage of a feeding habit by a mammal by forcing the intermediate host to go to a higher level.
So the cow, they say cows, deer, elk, they eat it. And then what happens? It goes into the gut tract?
It goes into the gut tract and then transforms into the adult in the small intestine and migrates up the bile duct into the liver. And it's a liver parasite.
How does it get out of the cow?
Well, it produces eggs.
Yeah, I see. And they come back out in the gut tract.
The eggs come out in the gut tract, and
then they start over.
Then they're eaten by a snail.
Oh,
the eggs have to embryonate, and then when they do, they embryonate outside.
Yeah.
And the snails forage on droppings of cattle, and they pick up the eggs. The eggs hatch inside of the snail, and then some very complicated. I'm going to say complicated biology, but all biology is complicated. So in this case, the egg then hatches into a myristidium, right? Mm-hmm. And the myristidium then develops into a sporocyst stage, which is a a stage which multiplied up from the meracidium. Okay, so the meracidium gives rise by multiplication to the sporozoite stage. The sporocyst stage, rather, I meant to say, the sporocyst stage then gives rise to another round of replication to make daughter sporocysts, which then produce cercariae. Because all trematodes produce cercariae, and all trematodes utilize snails as intermediate hosts. So how does the snail get the cercaria if it all takes place on land? These are not aquatic snails. These are land snails. So the snails in question here secrete sort of a mucousy, frothy material. Not a slime trail like you'd ordinarily think, but this is a product of their digestion. And the cercariae remain alive in the moist bubbles that are created by this excretion. And then the ants come along and they feed on these things and they get moisture from them, but they also get the cercaria, and then they get infected with the metacercaria.
They call them ant-appetising slime balls.
Yum, I'll have another one of those,
Isn't that great?
They are. They come in different
So these ants
flavors.
are amazing. They
are.
Grasp a flower petal, and they stay there overnight, but they detach the next day if the air temperature exceeds 18 to 20 degrees.
Sure, because they'll dry out.
But if the temperature is lower, they will stay there 7 days.
Yeah.
And when they don't defend themselves, but if they leave and come back, they come to the exact same place.
It's amazing. It's truly amazing.
The same petal.
It's amazing.
How would they know?
Well, they leave a trail. That's another great question.
Leave a trail.
No. They leave a specific trail. What is the genus name of this ant? Formica.
Oh, the ant. Sorry, I thought you meant
the ant.
Yeah.
The ant leaves a trail of formic acid.
Formic acid, yeah.
And they know where that flower was. They can just crawl right back up. That's how they find their way home.
Yeah.
They leave little trails of formic acid.
And the ant ingests the slime balls. The cercaria pierce the crop wall.
That's right.
And one or two invade and occupies the sub-esophageal ganglion. That's in the brain.
That's right.
Right?
That's right.
The rest of the cercaria insist in the gaster.
Exactly.
Which is the big butt of the ant, right?
Like a stomach. It's an enormous enlargement for processing food.
What happens to those encysted cercaria? Do they end up doing anything?
Yeah, they do. They end up as metacercaria.
Right.
So a metacercaria is a further developmental stage of a cercaria. Cercaria has a tail. Okay, it's like a little tadpole.
Yeah,
it looks just like a tadpole. And when it becomes a metacercaria, it loses its tail, and it secretes a cyst wall around itself as a protection against environmental change. And it can stay like that for quite a long time. Okay? It stays like that in the ant until something eats it.
Oh, I see. So the ones that are encysted are transmitted to the cow or whatever eats it and starts to cycle over. Yeah, and then
right.
Yeah, it's amazing.
It is, isn't it? It's truly amazing. I totally agree.
What a cycle.
Yep. And they call this the Master Manipulator only because it's easily observed. Okay, and everybody that's gone through a biology course in parasitology in the undergraduate level studies this parasite because it's so interesting.
Yeah, well, people are fascinated by this behavior manipulation.
They are.
It's really interesting. Now, there are a lot of interesting questions like, how does this parasite do this? And what's the temperature sensitivity, right? How does that work?
That's right.
So the thing that's cool here is they decided to look at this and take pictures using a micro CT scanner.
I haven't seen this before, so I went crazy when I saw it because you know me, I'm a visual person to begin with. This is absolutely stunning photography.
I wonder if the instrument is big. It just has
stunning.
It has equipment for looking at small things like these ants.
Well, it is. It's miniaturized over what the normal CT scan would be like for a human brain, for instance. And they got pictures of these encysted and unencysted metacercaria. Wait a minute, an unencysted metacercaria? How does that work?
Is that weird?
It's bizarre. It's not just weird, it's quite bizarre because the normal pattern is for this metacercaria to insist in a resistant stage, right?
Yeah.
And here's only one, located at the notch of the subesophageal ganglion because it's a double-bulbed portion of the brain of this animal. And that's where this particular unencysted metasecaria sits.
They say the oral sucker is in contact with the brain.
What does this remind you of? Alien.
I don't know how. We didn't do any CAT scans on the aliens.
No, we didn't. But if we had, we would have seen some bizarre...
So they take these CAT scans of these ants, which they collect in Alberta, and they ship over to London, where the micro CAT scanner is.
Exactly.
Have you ever seen a micro CAT scanner, Daniel? I know yours are.
They actually have these at a lot of research centers now. I'm sure we have a few at Columbia as well, and they're great because they have little I just call them containers or holders where you'll do studies where you want to look at, let's say, a rat or a mouse or whatever or model organism might be.
Yeah.
And the micro CTs are impressive. They in this paper they use the terminology a voxel. A voxel is really basically a pixel, but with that third dimension. So these voxels are a micron by a micron by a micron.
Incredible.
So the resolution is fantastic, and I think a lot of us are visual, Dickson. So these are the things that you make the cover of the magazine because you can create these wonderful 3D images.
I'll bet you could visualize a malarial parasite inside of a red cell this way.
Well, if you think about it,
why not? Sure.
Yeah, think about the size of your red cell. You're getting to the limit there, right? Because a red cell is about 5 microns?
5 to 7.
Yeah, and then you get down. So you're right at the edge. But then, why can you visualize it? Because then there's computer algorithms that allow you to extrapolate and create these images.
Exactly.
So this is actually something that is quite popular, and it's interesting here because as they talk about, there are two competing I'll say sort of approaches to how they think that this control occurs, and one is the mechanical, and the other is the neurochemical. And I don't think they're necessarily separate. I think they potentially both could be going on, despite what we're reading about here.
So they reconstruct these ants, and they have a beautiful photographs, right, where you can see the head and the thorax and the back part. What is that called again? The
gaster.
Gaster.
E.O. Wilson must be going crazy
and
for this.
You can see these eggs, but it's like the gaster is almost full of eggs. It's amazing. Well, I don't know how these ants are normal.
I mean Metasecaria.
Metasecaria. They look like eggs, don't they?
They do look like eggs.
They look like chicken eggs.
They do.
I mean, they are. I'm looking at this gaster, which is a big, if you think of gaster as the back part, the very back part of the ant, which is pretty big.
That's right.
And in this three-dimensional reconstruction, it is almost completely full
60 to 70 Metasecaria.
Amazing.
Per ant.
I don't know how the ant functions, frankly. If your gut tract were full like this, you'd have trouble. You'd be going to see Daniel Griffin.
Tell me about it.
And we'd be talking about you on TWiP.
You would.
And then, of course, you see in the brain, as you mentioned, an unencysted. Which is colored. Key. They colored, and
that is.
But they say there's a membrane around this, which is host derived, but the oral sucker is exposed. So why do these cercaria have oral suckers?
This is a metasecaria.
Metasecaria. Sorry.
Well, they have both an oral and a ventral sucker because when they are swallowed and they exist in the gut tract, they immediately transform into the adult trematode, which has an anterior and a ventral sucker. The anterior sucker, I must mention this, the anterior sucker leads to the gut, the esophagus, and then the esophagus bifurcates into two tubes, which are dead ends. They don't have an anus.
I see.
So they ingest tissue. In this case, they ingest liver tissue. And then the byproducts, after they've finished digesting what they want, they regurgitate back out into the host all of the stuff that they don't use. The other sucker helps in the attachment of this parasite to the host. And then in between both of those suckers, there's a birth pore, out from which come the eggs of this parasite.
I see. So they say that this SOG,
subesophageal ganglion,
would be perfect for manipulating locomotor activity and functioning of the mandibular muscles.
Right.
And they say there's an example of this in female jewel wasps. They inject venom into the cockroach heads, which targets the subesophageal ganglia to alter the host's drive to move.
Wow.
So Daniel, do you know about this subesophageal ganglia?
It's
a good place to target.
Yeah, no, it is a good place to target because this is sort of a central area from which they're going to suggest that this parasite is able to trigger this reversible tetany of the mandibular muscles.
Right.
As we described, not only do the ants need to go to this location, but then they need to stay there. And so the mandibles are basically going to lock down,
freeze up,
stay there.
That's right.
And so this SOG, as we'll call it, not only can control that, but it actually has wide-ranging effects on the neuro system of the whole ant.
Sure,
so it's a prime place to go if you want to control the ant's behavior.
Exactly. Can I introduce an anecdote here?
Yeah, of course.
About my
Our listeners usually like your antidotes.
Well, this is an antidote. No, it's not about ants. It's about some other insect, though, because part of my background is that I've had courses in entomology that I took while I was a graduate student here at Columbia back in the old days. Okay, so back in 1963, when I was a master's student here, I took 3 courses in entomology. One was called arthropod morphology, and another one was called insect identification, and then another one was called arachnid identification. So we had 3 separate courses given by one man named Dr. Roger Williams, and he was a wonderful person. He was a terrific teacher. And one of the exercises in arthropod morphology was to do a dissection. Now, what kind of insect are you going to give me that I can do a dissection on, right? Because most insects are pretty small. We had a jar. It looked like a mayonnaise jar for the 7th Regiment, filled with formalin, 10% formalin, and lubber grasshoppers. Now, a lubber grasshopper is huge. If you look them up on the internet, on the Google search, they're about 4 to 5 inches long. They're black with stripes along the sides of them. And so one of the options for this laboratory exercise that I was fascinated with was to dissect out the subesophageal ganglion of the lubber grasshopper. And I did it without breaking it.
Very good.
And I showed it to Dr. Williams, and he took a look at me and he said, "No one has ever done that before in my class." He said, "You've got good manual dexterity." Now, I must tell you that I was also interested in tying flies and stuff like this. So, an artificial entomology. The point is that you're looking at one of these turning points in your own career, and someone encourages you at that level. You want to take it to the next step, okay? And that's, I think, what kept me focused on parasitology. And, of course, lubber grasshoppers are not parasites. But just the act of trying to dissect out the brain of an insect was fantastic.
So having feedback was important for you. Right?
It was fantastic. He was wonderful at praising and not so good at saying, you shouldn't have done that. Really, he never said that. He would withhold overt negative criticism in favor of praising something when you did something right. And I think that's a talent that a lot of people don't have.
Yeah. So, Daniel, do you think that the Metacercaria are releasing chemicals that are influencing the ganglia?
That would be not what this paper is arguing.
They're
arguing is that there's a direct contact, and somehow this direct contact is controlling this central part of the neuromodulatory system of the ants. But I don't know. I'm always a little suspicious of mechanical explanations for things, and so at this point, I'm sort of left with this is interesting. You've got this neurosucker basically right here, potentially attached to the SOG, but I'm waiting for what sort of manipulation is it really that by being there it causes the SOG to produce these neuromodulatory substances, right? Or is it releasing them itself? And it's interesting to me. Is there an electrical? There's got to be some connection there. You can't just touch a nerve with your mouth and make it release, peptides and neurotransmitters. And so I'm left with, this is really interesting, and it's kind of eerie if you think about, oh my gosh, this parasite that's attached itself to basically the brain center of the fight and flight response of an ant.
Turned it into a zombie.
I hope I don't get infected with one of these.
It'd be very interesting to figure this out experimentally.
So what would be your next steps? Let's put the paper aside for a moment. Now, what definitive experiment could you think of that could answer whether or not it's chemical or mechanical?
Well, one of the problems is they say you can't infect ants in the lab, so you always have to go out and collect them. Right?
You think that's a problem?
Yeah, because what I would do is take the parasite and knock out individual genes and see which one blocks the behavior. But you can only do that if you can infect them in the laboratory.
Sure.
Or you could grind up the metacercaria and do a proteomic analysis and say what's here that could be neuroactive, right?
Right.
So maybe you could do that in the absence.
What about ablation with a laser?
Ablation of what?
Subesophageal area. Take a look at where this parasite locates.
Do that.
Now, could I replicate that with a laser ablation and say, if it's mechanical and I can do the same thing with a laser, the ant, then the mandibles now are locked in place. That's maybe the...
what's curious, though, of course, it's reversible, because if it's too warm, somehow it's changed. So the parasite must have a sensor that takes away the stimulus and allows it to walk back home and say, hi, I'm home. Because they say they have totally normal behavior when they go back to the colony.
Right.
This is a Disney movie waiting to be made.
I would think.
Totally. So, yes, I think it would be great if you could infect the... your experiment is good, but I would like to infect these ants. So I would work hard on developing a system to infect them. How would you infect ants with these, Dickson? You have to feed them these slime balls from the...
You have to keep the snails in culture.
Yeah.
And the ants in culture.
Want to do it?
Sure.
Yeah. I was thinking to use the technology they're using. Can you do the imaging in this cold environment where they're locked in, and then raise the temperature to, let's say, above that 20 degrees C, and then image and see, do they move away? Because if it stays physically right there and the physical relationship hasn't changed, it's a little harder to argue that it's the physical connection that's triggering.
It's a great idea.
Mm-hmm. Terrific idea. Yeah, that would be good. So one last thing, because we have to let Daniel go here. One last thing, they say it would be great to use these images to make big models to put in museums and get people excited. I think that's great, right? Because can you imagine a huge ant model with these parasites in its gut, and people would go, oh, in the brain. Oh.
Right. Do you remember the movie Microcosmos?
Nope.
Oh, that's going to be my pick of the week sometime on TWiP. Microcosmos was a fabulous film done with an amazing lens, which allowed you to take a picture in focus of a very small object and at the same time get the very large object in focus as well. So, for instance, they would take a picture of an ant, walking down a little path. Of course, for them, it was a big path. And at the same time that you're looking at the ant in focus, you can see someone walking in the opposite direction, a person. You can just see the foot of the person, and it's in focus. This was a special camera that they developed with a special lens. And they made a whole movie about using microcosmos. They showed lots of different things, and I think that if you typed it out and looked it for Netflix or something, you could probably find it. It's made by the same people, I think, that made Winged Migration, which is another wonderful film where there are no words. Basically, it's just looking at birds migrating from one place to another, but it's done in slow motion and up close and all kinds of wonderful nature films. That's where this would come in spades. I think this would be fabulous.
All right, Daniel, just before you go, can you give us a new case study?
Indeed.
And then when he leaves, you can do a hero because he's got to go.
Okay, fine.
Okay?
Well, that's okay. I'll work with you guys. I don't want to leave without hearing about the hero. Why don't we do the hero first?
Do the hero first. Okay, fine. And this is a hero that Vincent is going to love because Vincent is an Italianophile. And why? Because he's Italian. Come on, let's face it. And this really gives credence to the fact that if you know what you're talking about and you stick to your guns, eventually you're going to get credit. So this hero, his name is Giovanni Battista Grassi. Did I say that right, Vincent?
Yeah, it's good.
He's an MD, and his dates are from 1854 to 1925. So he's into the modern era of life on Earth. And here's what it says. Grassi was indeed a parasitologist for all seasons, working diligently on a plethora of biological systems, including invertebrate development. He worked on termites and honeybees. Parasitic life cycles. He worked on Ascaris lumbricoides, Strongyloides stercoralis, and Solastoma duodenale, filaria of various species, Hymenopus nana, Plasmodium vivax, and he's the co-discoverer, along with Filetti, of Plasmodium vivax. And then finally, he worked on vectors of human malaria and plant pathogens affecting wine grapes. So that's as broad and as wide an activity spectrum as you can expect from a scientist. Grassi was a thorough, observant investigator and made magnificent contributions to the advancement of knowledge in each of these fields. His worldwide fame arose from his research in collaboration with Amico Bignani, Giuseppe Bastinelli, and Antonio Dionisi. I didn't say that right. Dionisi. He identified the female Anopheline mosquito as the vector of human malaria. Grassi also discovered which species of Plasmodia cause which malarial fever pattern. That he was absolutely a famous man in all of his activities. So you might want to ask yourself, in the era of the advent of Nobel Prizes, why wasn't this gentleman and his group awarded the Nobel Prize? And it's a complicated answer, which relates back to his role in the laboratory with certain other people who came to visit and were insulted by him because he was a very abrupt person. And they went back to their own laboratories and they said, under no circumstances will that person ever win the Nobel. No, they didn't actually say that. But when the time came to choose the Nobel Prizes for the discovery of malaria, Grassi was omitted from that list. And instead, Ross was one of those people because he discovered insects transmit malaria. That was his contribution. And then Lavrin was part of that also because he worked on part of the life cycle for malaria. But they omitted Grassi, who deserved full credit because he discovered that only Anopheline mosquitoes transmit human malaria. And today he is now being heralded as a hero by me and by lots of other people too, because they realized that there was an oversight on the part of the Nobel Prize Committee. They should have awarded the prize to him. Or the next year they should have awarded it to him because they can only award it to 3 people, right?
That's right.
So the next year they should have given it to the 3 Italians that discovered the malaria parasites in humans. But they didn't. No. It was too bad. But he still is as revered a scientist as if he had won the Nobel Prize. So winning the Nobel Prize isn't the end-all for scientific fame and fortune. And this gentleman is living proof of that. So he's one of my all-time heroes as well.
All right. Giovanni Battista Grassi.
Nicely said.
All right.
I'll have another piece. With a little bit of wine, it's fantastic.
Okay. Now it's time for a new case study.
It is.
All right. I hope people enjoy this. This is a long, complicated, challenging case.
Another one. Wow.
And hopefully we'll give people enough time to write in. And this is the case of a male in his 50s who comes to see me. I say comes to see us, but he came to see me with concerns that he is infested with parasites. Now, he is a retired firefighter. He was a survivor of the 9/11 disaster, and he has been having several years of skin issues that have not resolved despite seeing multiple physicians, and he's referred to me actually by another infectious disease physician. He reports that all was fine even after 9/11 until we go back to 2009 when he is dating a Haitian girl, gets to know her, and then he actually goes down with her to assist in Haiti with the disaster relief efforts. While he's down in Haiti, he first notices this rash. He describes the rash as very itchy, it's worse at night, and he reports that prior blood work has consistently shown elevated levels of eosinophils. And he says that there was a prior biopsy that he's not able to get a copy of the report, but at that point the biopsy showed arthropods with compound eyes. He reports that in the past he's been treated with oral ivermectin, topical permethrin, and he also uses a topical clover sulfa concoction that he gets over the counter. He occasionally finds relief when he uses an application of ichthymol combined with diatomaceous earth and cornstarch. He describes ulcers that form, but these are preceded by small white objects which will then pop off his skin, and when he catches those, he can put them in water and they'll swim around in a circular motion. He's got some videos of those. He has had multiple other doctor's visits and repeat cystoscopies. That's where they scope your bladder. That was triggered by episodes of hematuria. He has had multiple biopsies and skin scrapings. The ones that we can get copies of all are unrevealing and just show either pieces of skin or nonspecific inflammation. He reports that before coming to see me, he had this episode where there were these forked-headed worms, and they actually came out while he was on the toilet, and he tried to photograph them, but then his phone fell into the toilet. This was followed by an episode where he became blind temporarily. And he saw another physician who suggested that maybe these symptoms were drug-related. So he is then sent to the lab with these pieces of skin scrapings, and he relates that when he went to the lab with the skin scrapings, the police arrived, there was a whole scene, and then he comes in to see me. And when he comes in, he's got the video we described of the object swimming around in water. He's got 2 fluid-filled bottles containing pieces of skin. He reports he's not allergic to anything. We get a little more social history. And in addition to the time in Haiti back in the early 2000s, he spent time in Egypt, where he swam in fresh water. He reports that he does have some issues with substance abuse, prior tobacco use, and he's unable to obtain a negative tox screen because he's unable to go any period of time without some degree of drug use. Family history is unremarkable. And on exam, he has these ulcerated areas on his neck, on his face, on his arms, his legs, and his scrotum. A CBC is done, which reveals an eosinophil count of 700. He has an abdominal X-ray, which show calcifications at the periphery of his bladder. We have some blood work. He's HIV negative, HDLV negative, 1 and 2. His schistosoma serologies are negative. Strongyloides are negative. A GI-PCR is negative. And we review all the available biopsies and scrapings, and all of these, including multiple stool O&Ps, are all unremarkable except for showing inflammatory changes.
Wow.
Okay.
How long did that case last, by the way? Just the physical examination. It sounded like pretty thorough examination.
Yeah, this was a pretty long discussion. Actually saw this patient several times to get all this information and outside records and lab reports.
Remind me again, how old is he again?
He is in his 50s.
50s?
Yes.
Okay. But was one of the early reporters to 9/11?
Firefighter.
He was a firefighter.
No, reporter, yeah.
He did survive 9/11.
Okay.
And then actually continued to work as a firefighter after that.
Wow.
If I had done that, I would see forked worms also.
In my dreams.
You know, there are lots of... We often get emails of people who feel they have parasites crawling out of their skin and so forth.
It's not unusual. It's not unusual. But this is, it's often the case where you could dismiss this and say, he's got a psychosis of some sort because of the trauma associated with having to deal with that incident every day, and then see a lot of his own firefighting mates die from cancers that they caught later on. But every now and then, something like this turns out to be real, and it's something that's never been described before. And if you don't pursue it, then you're never going to find out, right? It's a very perplexing, not too obvious on this one, Daniel. Nice going.
Okay.
Keep us thinking.
Before we go, let me just tell you one more thing. So, as usual, send us your guesses, right? TWiP at microbe.tv.
Absolutely.
This time, we will have some books of poetry for you.
Nice.
Sent in by listener Laurel. She's an author who
that's great.
Writes, and she has a book called Red Mother. It's her debut poetry collection. A love story told from the perspective of a parasite. Laurel sent me a couple of copies of the book, and we will give it to a winner next time, along with Parasitic Diseases 6th edition.
Which happens to be a red book.
It's a red book, that's right. So make sure you send your guesses to TWiP at microbe.tv. That's TWiP158. You can find it at Apple Podcasts, microbe.tv/TWiP, or on your phone or tablet when you listen to podcasts on your player. Just search for TWiP. Please subscribe so you can get every one as we release them. They're free. Why not get every one of them? And if you like what we do, consider supporting us financially. Go to microbe.tv/contribute. Have a number of ways you can do that. Daniel Griffin is at Columbia University Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Pleasure as always.
Dickson Despommier is at trichinella.org and thelivingriver.org.
Yeah, and we just put up trichinella.org.
Brand new.
Let's get those critical reviews coming in. We'd be glad to hear what you think of it.
Yes, Dickson and I revamped trichinella.org. It's now a modern website.
That's right.
And
that's great.
It
remastered all the videos.
Made me realize his huge contribution to trichinella biology. It's really quite remarkable.
It's very nice.
You should check it out. Thank you, Dickson.
You're welcome, Vincent.
I'm Vincent Racaniello. You can find me at virology.ws. Music on TWiP is by Ronald Jenkins. Thanks to ASM for their support. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
is parasitic.